have an afferent and an efferent arteriole kind of right here like this. This would be our little ball of capillaries. What do we call that ball of capillaries? Called it the glomerulus. Yeah, it's the glomerulus. And then it's surrounded by this capsule of epithelial tissue. What do we call that capsule of epithelial tissue? Bowman's capsule or the glomerular capsule, right? Either one of those works. And then we have this tube that kind of comes away. I've taken it and I've straightened it out, but what's that tube called? The proximal, PCT, proximal convoluted tubule, right? Again, we don't want to confuse it with the Pacific Crest Trail, so we want to make sure we call it the proximal convoluted tubule. We have this very unique structure that we see right here. That's the loop of Henle, followed by this, which is the what? The distal convoluted tubule, right? And then we have that emptying into a collecting duct, right? So we have a situation where things kind of look like that, right? And that was where we spent most of our time on Wednesday, was getting to know the anatomy of the nephron, and also looking at how those nephrons fit into the kidney. We know that the kidney has two parts, has a cortex and the medulla. So where's all this stuff found in the kidney? Is it in the cortex or the medulla? It's in the cortex, right? Yeah, indeed it is. Okay. So I feel like we're doing pretty good regarding the anatomy of the kidney right now. Are there any questions about that? Even if I feel like we're doing pretty good, maybe it doesn't necessarily mean we're doing pretty good, so we should check in. Okay. I assume we're doing pretty good. Okay. So let's go ahead then and talk about urine formation today. All right. How is urine being formed in this structure? Because we are a little bit light on time, I'm going to abbreviate this a little bit, but I still need to cover some of the main points with you, right? So urine formation occurs in three primary steps. Okay? The steps are drinking a beer and drinking another beer and drinking another beer. I'm just kidding. That's not the way that it works. Well, it is the way it works. But when you do that, there have these three steps that are happening in your kidney, right? Those three steps that are happening in your kidney are filtration, reabsorption, and finally a third process that we call secretion, okay? Or tubular secretion, but secretion works just fine. Okay. So we're going to go ahead and talk about these three steps and the way that they work today to be able to form urine. I think to some degree these steps might seem a little bit unnecessary. Like if you're looking at it, you're like, well, I could do that easily, right? And we probably could come up with a kidney that makes a little bit more sense than the way that ours works, because there is a little bit here that feels a little bit unnecessary. But we'll kind of talk about those when we get them. But I think that's why the kidney always feels kind of odd to people when they first learn it, is because there are some steps that you look at and you're like, well, that seems weird. Why would I need to do that? Well, we'll try to clarify those things as we get to them today. But really, at the end of the day, they actually do all make sense. All of these steps do. Okay, so let's go ahead first and let's talk about filtration. Okay, let's go ahead first and let's come over here and let's talk about our very first step, which is filtration. Okay, filtration is going to occur here in the renal corpuscle. So it's going to occur in the glomerulus and in that Bowman's capsule or glomerular capsule. So we're going to start the process of urine formation here by filtering our blood. So, you know, I think that most of you guys, I don't remember if I told both labs the exact same thing on Wednesday or not, but when you filter something, what you're doing is you are moving some things through a filter while you're excluding other things, right? And a great example of that is making coffee, right? When you make coffee, you filter, and you are basically excluding those coffee grounds based on their size, right? They're really, really big. They can't pass through the filter, but other things like water can pass through the filter with no problems at all, right? So filtration of our blood works in largely the same way. And we need a filter in order to be able to do that, okay? So let's go ahead and look at first at what the filter actually is made up of in filtration. Okay, so filtration occurs in the renal corpuscle. It occurs in the renal corpuscle, and the filter itself is made up of three primary things, right? So the filter has three primary components, I guess I should say. There we go. The middle of my board always lands in an awkward place. So the filter itself has three primary components. So let's go ahead and list those three primary components. Okay. The first is the epithelium of the capillaries themselves. Okay. So the first one is the capillary endothelium. I think I mentioned very briefly the other day that these capillaries are some of the few capillaries in our body that are fenestrated capillaries, meaning that there are fairly large gaps between the endothelial cells. Okay. So if we were to take a look at our capillaries of our glomerulus, so now what we're going to do is we're going to take our glomerulus and we're going to blow it up and take a look at a single capillary. Okay. What you would find when you look at those epithelial or endothelial cells that make it up is you find that there are fairly big gaps between some of those cells, right? So it kind of looks like this. Okay. We have our endothelial cells making this up, and we're going to see that there are gaps between them, right? So we have these fairly big gaps or fenestrations between them, okay? Remember, most of the capillaries in our body, if you remember back from our circulatory section, are what we call continuous capillaries, meaning that it's just a continuous line of endothelial cells. But here, we see these gaps or fenestrations between the individual cells, right? So we see that these are fenestrated capillaries. Okay. These are fenestrated capillaries. What this is going to do is it's going to create little tiny pores between the cells, but those cells are fairly small, and so when there's a gap between them, it means that the gap is also fairly small. We spent a little bit of time looking at blood under the microscope before our last exam, right, where we counted all of those white blood cells. Remember, most of our white blood cells, and even our red blood cells, are fairly big, especially if we compare them to these endothelial cells, right? So blood cells, especially, are not going to be able to cross through this membrane, right? So this is really going to exclude a lot of our blood cells from getting out, right? So what we see is that the red blood cells, right? The red blood cells that are kind of floating around in here, right? Whoops, sorry, I gave it a nucleus. The red blood cells that are floating around in here, and then also the white blood cells that are floating around in here are not going to be able to get out, right? These are fairly big. We'll make that a little neutrophil right there. We've got another one over here. We can make that another neutrophil. Maybe we can give ourselves a monocyte right over here or something like that. These big white blood cells aren't going to be able to cross through these little tiny areas, right? Think about your blood cells like the coffee grounds of your blood, right? They're not really supposed to get through that filter, okay? Okay, what do we know about epithelial cells? Do epithelial cells float in space? They don't, right? They have to rest on what? Some sort of a basement membrane. Very good, all right. If I taught you anything this semester, it's that epithelial cells don't float in space, right? They have to be on a basement membrane. So let's take a look at that basement membrane. It's called the basal lamina here, okay? And like most connective tissue, it's largely acellular. It's mostly just fibers of collagen that sort of make it up. All right, so here's our basal lamina, right? It's very fibrous. So that's kind of the next part of our filter here is the basal lamina. Basement membrane. Again, creates this kind of layer that we see here that kind of gives these epithelial cells a place to rest. And if you look at that, you say, well, it looks pretty continuous, but it's also fibrous, right? So little things, okay, if something is small enough to get through here, what we're hoping for is that it can find its way through those fibers to sort of get filtered out, right? It becomes a little bit more maze-like, but it does kind of end up there. One of the things that we need to talk about, and one of the things that we definitely want to stay in our blood and not get filtered out are plasma proteins, right? Proteins should not end up in our urine. We'll talk a little bit about that towards the end of lab today, okay? Most of your plasma proteins are positively charged, okay? They have a positive electrical charge around them. The basal lamina has this kind of acellular sort of structure to it, but it also has negatively charged glycoproteins as well. Okay, so it contains negatively charged glycoproteins. Okay, so you can imagine that in this basement membrane are all of these glycoproteins that have these negative charges associated with them, right? So the idea with having these negatively charged glycoproteins embedded in this basal lamina is that when a positively charged plasma protein kind of goes by it, we know that like charges repel each other. We know that opposite charges attract each other, right? So what happens is these negatively charged glycoproteins start to catch these positively charged plasma proteins, and they don't let them out of the blood, okay? So the basal lamina is really critical for catching and excluding those proteins from getting filtered out based on their electrical charge, right? So these exclude plasma proteins from being filtered. Based on electrical charge. Okay. So they exclude those based on their electrical charge, right? Size is only one criteria that we can use to filter things based on. We can also filter things based on their electrical charge. It just so happens that most of our plasma proteins have similar electrical charges, and as a result of that, we can create this sort of electrical barrier toward those things getting out of our blood. Okay, it's obviously not perfect, but it's pretty darn close, right? All right. So the last thing that we have here, okay, the last kind of component of our filter is we have these cells that cover our glomerulus. what are those cells that we saw on Wednesday that cover our glomerulus called? They're called podocytes, yeah. Remember, we said that name, podocyte, literally means foot cell, right? Literally, it means foot cell. So these podocytes are going to cover the Bowman's capsule, and they're going to create an additional layer of filtration that kind of occurs over those, right? So over all of this, and I'm about to make a big mess of my drawing, over all of this, you've got this thing completely covered by these podocytes, and these podocytes have these foot processes that kind of come off of them that kind of look like this, right? That kind of look like this. And what happens is that what they do is they kind of create these slits that kind of come off of them, right? I'm going to go ahead and kind of draw another one up here, kind of like this. This is another podocyte right here, and it's got that foot process that comes off of it, right? So you can imagine, or you can kind of visualize here, how what I'm doing is I'm creating slits between these foot processes, and that acts as an additional filter, okay? Does that make sense? So let's go ahead and add that to our list. Our third and final component of our filter are the podocytes. Covering the glomerulus. Okay. They create filtration slits. Okay, they create filtration slits. Does that make sense? Okay. So, when blood flows through your glomerulus, okay, what we end up doing then is we end up filtering the things that can make it through this filter, right? So we know that no blood cells are going to make it through this filter because of the fact that the capillary endothelium is fenestrated. There are these small gaps. They're not big enough to let blood cells through. We know that in a normal nephron, we're not going to have proteins making it through, okay? But generally speaking, other small stuff is going to make it through, right? So what does end up getting filtered out? Think about what else we find in our blood. What else would make it out of this? So what else do we find? So all of our formed elements, right? All of those are going to stay in our blood. But let's think about our plasma. What else is in our plasma that gets filtered out?
Water.
Water's a big one, yeah, for sure, right? So you're going to get rid of water, okay? So what's going to come out is we're going to filter out water, okay? But what else is in your blood plasma? Waste, to some degree, waste gets filtered out, right? Urea is fairly small. We do filter out a little bit of urea, okay? What else? What? Electrolytes, right? Yeah, right? We've got ions or electrolytes, right? So things like sodium, potassium, calcium, chloride, right? Just a couple of examples of things that might get filtered out. Okay, cool. What else? Is there anything else in your blood plasma? One thing we've talked about a lot today, or not today, we've talked about this all today, this semester is, what about glucose, right? We got blood glucose floating around, right? We have glucose floating around in our blood. You have amino acids. Those are the building blocks of proteins. Those can get filtered out, right? Anything small that we find in your blood plasma is likely at this point to get filtered out, all right? some of those amino acids have an electrical charge that would exclude them from getting filtered, but some of them would definitely be filtered out for sure. A lot of it depends on their electrical charge, but we'll definitely keep it on the list, right? All of those things are getting filtered out, okay? Now, there are some things on that list that are things that we're definitely going to want to keep. But before we get to that, let's talk about the force, the driving force of filtration, okay? So we have this happening right here, all right? But we always have to have something that sort of drives filtration. I know that we've talked about diffusion a lot between Bio 1 and 50A in this class. And we know, probably by now guys can do this one in your sleep. Diffusion occurs under a blank gradient. A concentration gradient, right? What kind of a gradient do we need for filtration to occur? You guys know? It's not concentration. What kind of a gradient do we need for filtration to occur? Well, let's think about coffee for a minute, right? I'm simple. I drink drip Folgers. It's not exciting, but I'm also cheap. I spend all my money on bikes. I don't spend it on coffee. So I'm cheap, and I'm cheap in one way. I'm not cheap in another way. I'm broke in other ways. But sometimes you might want to make coffee where you have a French press, right? What are you doing when you press that coffee to make that when you do the press? You're applying pressure, right? Filtration occurs under a pressure gradient, okay? Now, with drip coffee, okay, what provides the pressure? I like it. That just makes the coffee taste good, right? You can have cold brew coffee though, right? Yeah. It's a little different, I know. But there you go, gravity, right? With drip coffee, gravity applies the pressure, right? When you do a French press, right, you apply the pressure by pushing down on that plunger, right? I'm not so uncultured, I don't know how a French press works, thankfully, so that's good. Okay, anyway, so pressure, right? We need to apply pressure. So that means that there needs to be a pressure gradient in here that is going to allow filtration to occur, right? Okay, so let's talk about that a little bit. Is there any pressure that we can think of in the system? Say yes. Yes, there is, right? Because in the blood vessel, that has blood pressure. Say blood pressure. Blood pressure, right? Yeah, there's blood pressure in those blood vessels, right? So we know that it isn't a lot in our capillaries, but there is blood pressure in our capillaries, and that blood pressure pushes out or in? Out, right? That blood pressure is pushing out, right? So we have blood pressure, or what we called hydrostatic pressure, that was pushing out, right? So one of the driving forces for filtration is simply the hydrostatic pressure that pushes out, right? So the driving force for filtration here, right? Driving force or pressure for filtration. Part of it is hydrostatic pressure. Okay. In your capillaries, your blood pressure is around 55 millimeters of mercury. Okay, it's around 55 millimeters of mercury. So it's pretty low at this point, but it's around 55 millimeters of mercury. Okay. Now at the very end of our unit on the circulatory system, we talked a little bit about blood flow through capillaries, and we talked about the forces that affect those. Hydrostatic pressure was one of them. Was there another pressure that we see affecting capillaries and the movement of fluid? osmotic pressure, right? What was osmotic pressure caused by? By what? Sorry, I'm deaf. Proteins, yes, thank you, right? Colloid osmotic pressure was due to proteins. And was that trying to push fluid out of the capillaries, or was it going in the opposite direction? It was going in the opposite direction, right? It was pulling the fluid in, right? So that's here as well. We can't escape that. So we have colloid osmotic pressure as well. Those pesky plasma proteins creating an osmotic pressure gradient here, right? So we have that, okay, and that moves the fluid in the opposite direction, okay. That tries to move the fluid in the opposite direction. It essentially opposes blood pressure. Fortunately, it's only about 30 millimeters of mercury, though. I have that right? I do. Okay, so I'm going to put a negative here, just to show that it goes in the opposite direction, right? So I made this a positive number, meaning that it favors filtration. I made this negative because I want it to show that it's going in the opposite direction, right? Okay, so we have two pressures here that are important. There is a third pressure, all right? There is a third one here, and that's because we're excreting this fluid. We're excreting this fluid into a really small space, right? And the other day under the microscope, we saw that glomerular space within the Bowman's capsule. Remember seeing that little open space around your glomerulus? Guess what? That's also filled with fluid, okay? And because it is a fluid-filled space, it means it's also exerting outward pressure. Okay, so the fluid in the Bowman's capsule is also pushing pressure on here, and it's fighting that hydrostatic pressure as well. Okay, so it's the exact same kind of idea that we see happening with the colloid osmotic pressure. Okay, so we have a third pressure, and that one is also pointing in this direction, opposing filtration, and that's just something we're going to call the fluid pressure in the Bowman's capsule. Basically, it's blood pressure, but not in the blood. It's in the Bowman's capsule. The capillary isn't the only place that gets its pressure. Okay, and again, that's going to oppose filtration. That runs about 15 millimeters of mercury. So it's very, very low, but it is there. We're going to go ahead and do minus 15 millimeters of mercury here. Okay, minus 15. Okay. So if we do the math on this, okay, if we do the maths on this, we have 55 millimeters of mercury minus 30 millimeters of mercury minus 15 millimeters of mercury. If I can do maths correctly, and I think I can, that comes out to 10 millimeters of mercury, am I right? I'm not joking, am I right?
Yeah.
Okay, thanks. All right, 10 millimeters of mercury, okay? There we go. I have an existential fear of doing math in front of groups of people, so that just came out. So what that does is it gives me a driving force of 10 millimeters of mercury that drives filtration. It's not a lot, but it's enough to be able to call filtrate, okay? To be able to create what we call filtrate, okay? And what we're filtering out are the things that we have listed here on the board, okay? Any questions so far? Yeah. Well, there's glycoproteins embedded in the basal lamina, and they catch plasma proteins. Yeah. Okay. All right. Any other questions? Okay. So this blood is flowing through our glomerulus. We're filtering out the things that we have listed right here. Okay. You're not filtering out all of your blood plasma. You're not filtering out all of your blood plasma at once, right? What that would do is it would create a situation in that efferent arteriole where you're just sending like a thick slurry of blood cells into there, right? And that doesn't work, right? So we're removing some of our blood plasma here, but not all of it, not even close. Only about 20% of your blood plasma is getting filtered out at once, okay? In the course of a minute, okay, what do we say the other day? That over your kidneys receive approximately 20%, 20 to 25% of your cardiac output, right? Let's do a little bit of math on that, right? What's your cardiac output at rest, roughly? About 5 liters a minute, right? Okay. So if your kidneys are receiving 20, let's make it easy, 20% of that 5 liters. How much blood is going to your kidneys every minute if they're getting 20% of that 5 liters? A liter, right? Okay. That's 1,000 milliliters, okay? If you're filtering out approximately 20% of your blood in any given minute, right, to make this filtrate that we talked about, that's going to be about 200 milliliters per minute that you're making, right? That's a lot, right? 200 milliliters per minute. Let's do a little bit of math on that, okay? You are filtering out 200 milliliters per minute. So that means in 5 minutes, you filter out a liter of blood plasma, okay? And 60 divided by 5 is 12 time, that's 12 liters an hour that you're making. Am I right? Did I do that right? You're making 12 liters of filtrate every single hour. That's a lot, right? Over the course of 24 hours, over the course of a day, you're making 288 liters of filtrate. 288 liters of blood is getting filtered out every single day. If we assume that your cardiac output is 5 liters per minute, do you make 288 liters of urine a day? No, not even close, right? Do you even have 288 liters of fluid in your body? No, not even close, right? You would have dried up and shriveled up a long time before you get to that, right? We only produce about 1 liter or 2 of urine a day, right? So what that means is that the vast majority of the blood that we filter out has to get put back into the bloodstream as we continue to make urine, okay? That's where our next process comes into place, is reabsorption, okay? So we definitely are going to have to reabsorb water, okay? We're going to have to reabsorb like 99% of the water that we just filtered out, right? A significant amount of it, okay? But there's other things in here that we probably kind of want to keep, right? Glucose is a really good example of that, right? Like, I mean, presumably, as an animal, I worked really hard for that glucose that's floating around in my blood because I got it from a meal, right? And meals, we all know, don't come easily. Humans are kind of an exception to that. But other animals, they have to work for their meals, right? They have to work really hard, and those meals aren't a guarantee. So the last thing that you want to do is you probably don't want to be urinating away all of that fuel that you just got, right? So we definitely are going to want to reclaim this, okay, if we can. And then amino acids are the same way. Those probably came from a meal as well, and we can use those to build proteins. So we don't want to get rid of those, right? So they were small, they didn't get caught by the filter, but we definitely are going to want to reclaim those things in the process of reabsorption. Okay? Ions as well, right? Some of these we can get rid of probably, but we don't want to mess up the ionic balance of our body fluids too much either, because that's how action potentials work, right? And if I'm just willy-nilly urinating away sodium, guess what? My action potentials aren't going to do what I need them to do, and we all know how that goes for us, right? It doesn't end well. So most of this stuff we really kind of want to keep, right? So we need to sort of figure out like how we're going to do that, and that's the next step of reabsorption. Just one really quick note on filtration before we move on. I know we took a while to get there, but at the end of the day, it still is blood pressure that drives filtration, right? That is the primary driving force. There are a couple of things that oppose it, but at the end of the day, it's still blood pressure that drives it. We might think that somebody with high blood pressure would have a higher rate of filtration, right? That would be a reasonable conclusion, right? If your blood pressure is higher, then in theory, this number is a little bit higher, and then if the math all works out the right way, this number would also be higher as well, right? That would be a reasonable conclusion. It turns out that that's not actually the case. It turns out that over a pretty wide range of blood pressures, we're able to keep the rate of glomerular filtration pretty stable. And that has to do with an anatomical feature that we call the juxtaglomerular apparatus. We looked at this very briefly in lab on Wednesday. Okay. Do you remember what the juxtaglomerular apparatus was? Besides a cool name. Remember, our nephron looks a little bit different than this, right? We've got our Bowman's capsule right here. We've got our proximal convoluted tubule. We've got our loop of Henle. And then that distal convoluted tubule, remember that what it does is it actually wraps really close to our blood vessels, right? Let me actually do this slightly differently. I want to go the other way with it. All right, it comes around, it gets all convoluted and weird, and it kind of comes around like this, and it kind of does this business before it dumps into a collecting duct, right? So the idea is that distal convoluted tubule, right, passes really, really close right here to our afferent arteriole. Remember, in your distal convoluted tubule, you had a group of cells called what? The macula densa cells, right? And in your afferent arteriole, you had a group of cells called what? Remember what those were called? They were called juxtaglomerular cells. They were also called, what were they called? Granular cells, okay? They were also called granular cells, all right? So what happens is that with fluid, if blood pressure does increase, right? If blood pressure does increase, what happens is that the flow through the distal convoluted tubule is going to increase, okay? The macula densa cells in the distal convoluted tubule are going to talk to the afferent arteriole, the granular cells, and say, whoa, 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 flow is way too high, we need to slow it down. So what does the afferent arteriole do? It constricts a little bit, okay? And what that will do is it will slow down the rate of glomerular filtration, and the glomerular filtration will come back down. If the flow through the distal convoluted tubule gets too low, the afferent arteriole will relax, it will get bigger, it will allow more blood to flow into the glomerulus, and the rate of filtration will go up, okay? So the distal convoluted tubule is always talking to the afferent arteriole through a process called ready for this one? Tubuloglomerular feedback, okay? The juxtaglomerular complex what we see is something called tubuloglomerular feedback. tubuloglomerular feedback. It's a funny word. It's a big, long word. It makes sense, though, right? Tubulo is referring to the renal tubule, and specifically the distal convoluted tubule. And glomerular is referring to the glomerulus. So those two things are talking to each other, and that's where the macula densa and the juxtaglomerular cells come into play. Okay. So over a wide range of blood pressures, we actually see that glomerular filtration stays relatively constant because of this. And that benefits us, right? Because if this wasn't in place and glomerular filtration did go up with high blood pressure, what we would probably see is that patients with high blood pressure are chronically dehydrated, right? Because they're just making a ton of urine as a result of that, right? And we don't necessarily want that. So this helps keep you from losing excess water if you have high blood pressure. We're probably all dehydrated as it is. We don't need to lose any more water. Okay. All right, let's go ahead and talk a little bit about reabsorption then. I don't have as much to say about reabsorption. We'll see if we can finish that before we run out of time. And what we don't finish here in lecture this morning, we'll have to finish in lab today. Let's go ahead and kind of move on here. Let's go ahead and talk about reabsorption. Okay, so reabsorption. Reabsorption is the movement of material from the renal tubule into the blood that surrounds it. And if you guys remember your anatomy from class on Wednesday, remember that when this efferent, get rid of this business. Remember when this efferent arteriole leaves, right? Remember when this efferent arteriole leaves, right? What it does is it forms these capillaries that completely surround, right? They completely surround the entire renal tubule. What were those capillaries called? Do you guys remember? They're called the they're called the paratubular capillaries, right? They're called the paratubular capillaries. But specifically around the loop of Henle, they're called the vasa recta, right? We'll talk a little bit more about the vasa recta later on today because it's a really, really cool structure. But these paratubular capillaries completely surround this. So when we move things from the proximal convoluted tubule, say, to the blood, remember there is blood right next to this, right? We're keeping really close connection between what's inside of the renal tubule and the blood, okay? So we have that happening, right? So the reabsorption here, okay, is basically we're moving things from movement from the renal tubule to the blood, okay? So we're moving things in that direction, from the renal tubule to the blood. Remember, we just said you're making anywhere, depending on the math that you do, you're making anywhere from 180 liters of filtrate a day to over 200 liters of filtrate a day. Okay? So we have to reabsorb about 99% of that fluid that we just filtered out. Okay? So we reabsorb about 99% of our filtrate. Okay. Turns out that it's easier for us to selectively reabsorb the things that we want to keep than it is for us to selectively filter the things that we want to get rid of. So it kind of works out a little bit here, okay, for us. It ends up being a little bit easier, even though it seems a little bit kind of counterintuitive. Of that 99% of filtrate that's reabsorbed, about 75% of it happens in the proximal convoluted tubule, okay? So we're reabsorbing most of it almost immediately, okay? Remember, this is helped because of what feature of the proximal convoluted tubule. What did the epithelium of the proximal convoluted tubule have on it the other day? Microvilli, yeah. So thanks, you got that right. That was my miming, my pantomiming of this, right? The microvilli. Remember that the cuboidal epithelium in the PCT is covered in microvilli. So what that does is it really increases the surface area of the proximal convoluted tubule, and it makes it easier to move that massive amount of fluid across the membrane of the proximal convoluted tubule. Okay, so most of this is happening in the proximal convoluted tubule. There is a little bit that happens a little bit later on, but most of it's happening in the proximal convoluted tubule. And most of what's reabsorbed is water, okay? We have to get this water back into our bloodstream because if we don't, we're going to become dehydrated exceptionally, right? So most of it is water. The way that we reabsorb this, okay, most reabsorption takes place because of the active transport of sodium. Okay. So the way that we reabsorb most of this is through the active transport of sodium. What does it mean for transport to be active? You have to use ATP, right? Yeah. Active transport involves ATP, right? So here, what we're doing is we're actually moving sodium against its concentration gradient from the renal tubule into the blood, and we're using ATP to do that, okay? But what that does is that creates an osmotic gradient that pulls water along with it, right? So it's done by so we actively transport sodium across, and then water follows by osmosis. Okay. Water follows by osmosis. Okay. So we can't drive that water across unless we're using energy to reabsorb that sodium. Turns out that the sodium active transport also drives the reabsorption of glucose and amino acids and things like that as well, right? So we see that the glucose, amino acids, etc., also follow sodium. Okay. So we can't really absorb most of those things without sodium. So we're reabsorbing most of that fluid back into our renal tubule. All right. I don't have a whole lot more to say about that. By the end of the proximal convoluted tubule, though, you definitely have reabsorbed most of it. The fluid that passes into the descending limb of our loop of Henle, if we have about 200 liters of fluid a day here, we're down to about 50 liters of fluid a day here. So like I said, about 75% of reabsorption happens almost immediately, okay? Okay, so that actually kind of puts us in a good position, and it gives us to a good stopping point. If there's no, I'll ask in just a second if there are questions about this before we stop. But what we need to do then is I need to talk to you guys a little bit about the loop of Henle, okay? Because why the heck is it there, okay? And then what we'll do is we'll finish up with the distal convoluted tubule and the collecting duct in there.